Results I'm going to describe is based on collaborative work in my group at Wayne State University, the Shinazi group at Emory University, and Co-Crystal Pharma. And since we are switching from HIV, from dengue to Zika, and now norovirus, I'm going to give a very brief background regarding norovirus, which is a non-envelope positive strand, single-strand RNA virus. So first we are going to briefly discuss epidemiology and the clinical course and the pathology, and then some structural and molecular aspects. I like to emphasize the recent advances such as recombinant norovirus proteins and structures, cell-based assays, and animal models, which are critical for the development of antivirals and vaccines. Then we'll focus our attention on the two drug targets, the protease and the polymerase. Very briefly, we'll discuss clinical trials, and we'll conclude with animal models. So I'm going to do my best to cover a wide range of topics over 20 minutes. The hallmark of the disease is watery diarrhea and vomiting. Therefore, norovirus is described as a two-bucket biological agent. And there are about 700 million infections worldwide, resulting in about 200,000 deaths. In the US, annually there are about 21 million infections and resulting in about 800 deaths. And the CDC estimates that the average person will experience 4 to 5 episodes of norovirus infection during their lifetime. The biggest concern is regarding children, immunocompromised individuals, and the elderly. Currently, there are no antivirals or vaccines. And the disease occurs in enclosed spaces. So this is in retirement centers, nursing homes. It can be restaurants, catered meals, schools, daycare centers, and vacation settings. The main modality of transmission is from person to person, and there is also foodborne. It's challenging that since this is a non-envelope virus, it's and with particular structural features, is very difficult to disinfect, and 20 virus particles are sufficient to produce an infection. I've talked about the exposure already. The incubation period is about 24 to 48 hours. We discussed the symptoms and the outcomes. So in a healthy individual, the infection is self-limiting. There is a concern regarding infants, young children, elderly, and the immunocompromised individuals. A number of treatments have been tested. None of these are satisfactory, and therefore antivirals and vaccines are needed. Regarding the pathology of the disease, these are the villi from healthy volunteer prior to norovirus challenge. And after norovirus challenge and during gastroenteritis, there are these characteristic broadened and flattened villi. this is the same experiment in a pig, scanning electron micrograph of the villi prior to norovirus infection and after norovirus infection. So there's this shortening, blunting, fusion, or even absence of villi. So there is a very clear pathology here, and how the virus damages the intestinal tract. And when we think about antivirals, there are a number of intervention scenarios. So for example, first and foremost, treating immunocompromised patients with chronic norovirus infection, and these individuals can be infected for weeks, months, and there are also cases for 1 or 2 years. So this is in these patients, the course of the infection is very long, and they would be the first candidates for therapeutic intervention. Another modality would be preventing, reducing norovirus shedding in acute infected patients. And a third therapeutic intervention modality could be prophylactic in cases of norovirus outbreak. This is an electron micrograph of norovirus particles, and this is the crystal structure of the norovirus shell solved at 3.4 angstrom resolution. And that is this VP1 protein that has 3 particular regions, and perhaps from a vaccine design point of view, this P2 region is the most important, and I'll come back to this later. The concern is that the virus evolves very rapidly, and there are going to be changes in this the major viral protein, that's viral protein 1. Very briefly, a description of the calicivirus genome, so in the calicivirus family, there are 5 genera, including norovirus, and the 5' encodes for the non-structural genes, and the 3' end for the structural genes, such as VP1 and VP2. And in some caliciviruses, there are 2 open reading frames. In norovirus, there are 3 open reading frames. And this is the organization of the genes. The black arrows represent the norovirus protease cleavage site. So that's the NS6 protease. And we are focusing on the norovirus protease as a target, and since this enzyme and the polymerase are essential for the life cycle, the light arrows represent cleavage sites that occur in some of the caliciviruses, but the black arrows represent conserved cleavage sites across the calicivirus family. Very briefly regarding the classification of noroviruses, there are 9 genogroups, and each genogroup has genotypes. And we are focusing on genogroup 2, which represents about 90 to 95%, and genogroup 1, which represents about 5 to 10%. And pigs are a good animal model, so this genogroup 2 infects both humans and pigs. And I'll come back to this when we'll discuss the various animal models. And very briefly, I'm going to describe the life cycle. That is in some animal noroviruses, the receptor has been identified. In humans, it hasn't been characterized yet, but it's known that sugar moiety is important for binding of the virus to the cell surface. And so this is a plasmid RNA virus. The RNA is translated and then cleaved by the viral protease, and then the replication of the genome proceeds and virus particles are produced. And so the protease and the polymerase are logical steps for therapeutic intervention. This is the crystal structure of the norovirus protease. In blue is the N-terminal domain, which is a twisted beta sheet, and in red is the C-terminal domain. And between the two domains, that is the active site, and those are the three key residues. This is a cysteine protease, and our goal is to design covalent inhibitors that are going to completely inactivate the cysteine. This is the surface representation of the protease structure. Again, in red, the catalytic triad. The dotted line represents the binding of the viral polyprotein, and the various pockets represent the S1, S2, and S4 pockets represent the binding of the polypeptide chain. S3 is not well defined. It's the S3 pocket that is very shallow. These are the cleavage junctions, and I'd like to focus your attention on P1 and P1 prime. The P1 is glutamine or glutamate, and the P1 prime is a small amino acid residue, glycine or alanine. So when we think about structure-based drug design, we want to mimic the cleavage product, and on top is the cleavage product of the genogroup 1 norovirus protease, and the bottom is genogroup 2. And based on these cleavage product, we designed, synthesized, and tested a number of compounds. Currently, our lead compound has a 90 nanomolar activity in a norovirus replicon assay, and 200 nanomolar activity in the enzyme assay, and a favorable toxicity profile. We assay the activity using a FRET assay. This is the substrate. When it's cleaved, then we measure the increased fluorescence, but based on this assay, we evaluate the IC50. So this particular compound has 50 nanomolar enzyme activity. When we think about medicinal chemistry, conceptually, we can evaluate our molecules based on a backbone, P1, P2, P3 side chains, and the cap. And last but not least, this is the warhead. So our goal is to bind the inhibitor covalently to the catalytic cysteine residue. And the medicinal chemist, Frank Amblad, takes the lead regarding the synthesis of compounds for the project. we evaluated a number of warheads, and in addition to optimizing the side chains, we looked at the binding of the warhead to the cysteine, which is located here. And it turns out that aldehydes provide a perfect orientation and positioning of the inhibitor for reactivity for the cysteine. When we looked at this microreceptor, the warhead points in the wrong orientation. These are the modeling studies. Currently, we are working on crystal structure to validate these observations further. So to summarize this part of the talk, we have 3 norovirus compounds with low nanomolar activity. Aldehyde-based compounds are the best. Mechanism of action is irreversible inhibition of norovirus protease, favorable toxicity profile, CC50 ratio over 1,000, favorable ADME toxicity prediction 2 to 4 star rating, so we want something 5 or lower, and we also perform specificity studies. So the next step is animal studies in collaboration with the Wolbus lab at the University of Michigan. Now I'm going to switch to the other drug target, the RNA-dependent RNA polymerase, and this is the crystal structure of the APO protein, and this is the structure in various orientation for the complex. This structure has a classic right-hand structure, so the finger and the palm are rigid, and the thumb is flexible. A number of compounds have been identified by the Shirazi group at Emory and Peter White's group in Australia. Among the nucleoside analogs, 2'-C-methyl C has the best activity, 1 micromolar. These non-nucleoside inhibitors have been characterized based on screening of about 20,000 drug-like molecules, and currently compound 02 has a certain micromolar activity, so this is a starting point for optimization. Very briefly about vaccines. So I described to you the crystal structure of the protein, and these virus-like particles are being used for vaccine development. So there's a bivalent vaccine that contains genogroup 1 and genogroup 2 norovirus, and this is currently in phase 2 phase 2B clinical trials, and this is a bivalent intramuscular vaccine, and since there is no RNA in the particle, it is incapable of reproducing and cannot cause infection. So this is just a recombinant protein that assembles to form the shell. There is another vaccine in clinical trials, and this is a tablet rather than an injection. The challenge with vaccines is that the norovirus evolves very rapidly. So there is this figure, and I'd like to focus attention to the arch at the top. This is genogroup 1, and this is genogroup 2. So those are quite different, and most likely vaccines will require an annual immunization, something similar to the influenza vaccines, so since that is very rapid evolution. Among animal models, the immunocompromised mouse model is the most promising, and the gnotobiotic pig is the ideal model. So the low-cost model is used initially for antivirals, and then one can proceed to the high-cost model, and the same approach is used for vaccines. And before I stop, I'd like to acknowledge members of my group at Wayne State University. Ben Kuiper is a PhD student who took a leading role in this in terms of cloning, protein inspection, purification, enzyme assays, crystallographic studies, and drug design approaches. This is a collaboration with the Shinazi group at Emory University. Frank Amblad is the lead medicinal chemist on the project. Peng Liu and Sherman Zhou are also chemists. Later Bassett contributed with replicon and cytotoxicity assays, co-crystal pharma. Crystallographic data are collected at Argonne National Laboratory, and the next step is going to be animal studies in collaboration with the Wobus lab that has this immunocompromised model. I thank you for your attention, and I'll be happy to answer any questions you may have.